my pleasure to welcome you to the last of our Tuesday night lecture series, but certainly not the least. And it's my pleasure to introduce Professor Stephen Hurd, sitting right down here. Stephen did his PhD work 1993. Let me think about this a minute. University of Pennsylvania.
Very good, very good.
I think under the direction of Robert Rickless, if I recall correctly. I don't have that actually written down. He then went on and did a postdoc at British Columbia, where you worked with Dolph Schultz. Shooter, yes, Shooter. Yes. Okay, another interesting person working on sympatric speciation. Then, in 1995, he came to the University of Iowa, and I must say that one of the stars on my CV is that I got to chair the search committee that had the pleasure of recruiting him for our faculty at Iowa. And so we were colleagues for about 7 years. During that time, we did some work together. We became good friends. We drank a significant amount of beer together, I believe.
Highly significant.
Yeah, it's a highly significant amount. And then in 2002, he got a golden opportunity to move on to the University of New Brunswick in Canada, back home in Canada, where he is now professor and chair of the department, and you're doing some sort of administrative dean thing.
I'm not chair anymore.
You're not chair anymore. So he, well, we'll talk about the implications of that. Right. So he obviously is an important person in their administration of that university and those science departments. He teaches courses in general biology, entomology. I've seen some of his tests. They're hard. Ecology and evolution. And his research has been, one of the things we liked about it from the very get-go when we hired him was that it was hypothesis-driven research. And so Steve is constantly testing hypotheses, and he has focused primarily on looking at the controls of the biodiversity that we have on the Earth. And tonight he's going to talk about diet specialization. And this is the kind of lecture you don't want any children at, because he's going to talk about the importance of being a picky eater and not something you want to encourage as a parent. So please welcome Steve Hurd.
All right, so Steve gave you a little bit of Capsule Bio, and that's partly on here because I do have the little Iowa logo on there, but I also want to remind myself here to make a couple of acknowledgments. So most of the funding is either from the NSF, which you all know, or its Canadian equivalent, NSERC, which is our national science funding organization. And over the years, and I'll talk about work here that goes through about 12 years of history. Some of it has been, well, indirectly with Steve, but directly with John Mason, who's now at Iowa State, and a large number of students and so on. So it's not all mine. And what I want to do is I'm kind of tell you a story. Steve just told everybody I'm a good hypothesis tester, but I'm actually going to tell you, I'm going to be a storyteller today. I'm going to take you through a bit of an evolution of my own thinking about this study system I've been working on, which I'll show you in a minute, and how I've seen the same field of plants sort of differently over the years, and why I think some of that's interesting. So that's a bit of a vague thing, but you'll see what I mean. So let's start with, you've probably all seen versions of this little thing before. This is the Speciescape graphic. And well, this is just a little sketch that scales the size of the little pictures by the diversity of the group it represents. So the point it's making is that virtually everything that matters is an insect, and everything else doesn't matter very much. There are about 1 million insect species described worldwide. We probably have 3 to 30 million total, but nobody really knows. And compare that, so that's that huge June beetle up there. Compare that with about 10,000 bird species worldwide, or about 5,000 mammals. There's the little elephant there. And we know virtually all the birds and mammals, so that's all we're ever going to have. So insects rule, and they rule not just in biodiversity, but in terms of the number of individuals or in terms of biomass. But to me, diversity is sort of the interesting problem. I'm interested in why we have so many insect species in the world. And it's a ridiculous number. I mean, we're talking millions of these things. Among the insects, so to narrow it down a little bit, among the insects, some of the groups that are particularly diverse are the herbivores, the insects that feed on plants are astoundingly diverse, and parasitoids, which I'll talk about a little bit later if you don't know what those are. are also very diverse. And in fact, about half of all animal life on the planet is made up of either plant-feeding insects or parasitoids that attack them. Many of them are picky eaters, so they're extreme diet specialists. And so the obvious question, and I'm not the first one by any means that asked this, is whether that kind of diet specialization can explain the evolution of this remarkable diversity. Do we have so many insects because each one of them eats just one plant, or sometimes one organ or tissue of one plant? And so we're trying to get at that question. Now, this isn't my work here, but this is sort of a well understood case of fairly recent evolution of a new insect through diet specialization. This is the apple maggot fly. It's a pest of apple orchards in North America, but it's a native North American fly that, before apples were brought over here, attacked hawthorn and it laid eggs, and the larvae developed in the haws. With the cultivation of apple in North America, starting maybe 250 years ago or so, we had a host shift where these flies started attacking apple instead. In that time, and it was first documented in Michigan in the mid-1800s as something that was a problem for your apples. And since then, we know that it has evolved differences in host choice behavior, in mate choice behavior, in timing and development that suit one genetically distinct strain or race of flies to attacking apples and another one to attacking hawthorn. So we now have these two so-called host races. This is a historically documented case of new biodiversity. They're not yet distinct species, but they're along that road. And we know a lot about this example through work by people including Guy Bush and Jeff Fetter, Sir Verlocker, and a bunch of names you may or may not have ever heard of. We know a lot about this case, but it's a bit of an annoying particular case to be our test case, because this is the only thing that eats both hawthorn and apple. Or at least as far as I know, it's the only insect that eats those two plants. So it's pretty hard to use this system to ask how common and how predictable this kind of evolution is. I mean, we know it happened to this fly, but how do we go much further than that? And so we've been working for 15 or more years now with this system, which is goldenrods. And so we want to know, if we're studying goldenrods, we can get some handle on whether this kind of evolution of new diversity by host specialization is just a weird, wonderful thing that happened in apple maggot, but it's not really important in the big scheme, although it's pretty cool, or whether this is actually something that's common and happens all the time. Now, the first thing I want to say about this field of goldenrod is that when you look at that field of goldenrod, your first impression is, okay, there's some goldenrod. And I'm going to show you sort of as we go tonight that we can find more and more finely in this field and see interesting structure and interesting variation in there. And the first thing I'll say is that that's not a field of one goldenrod, it's a field of two different goldenrod species. And many of you will know them and some of you won't know them. And Steve was kind enough to bring me one of each for you today. And so I'll pass them around. There are two goldenrod species on the next slide. I actually have a better picture. But I'll pass these two around. And notice the difference in hairiness of the stems is the primary difference. You'll also notice the sort of golf ball swellings on the top, and I'll get to those later. If you don't, most of you will know what those are, but for those of you who don't, I'll get to that later. So here are my goldenrods, and here are the two species I'm now passing around. The hairy stemmed one, which is late goldenrod, Solidago altissima, for those of you who prefer the Latin names. And this is the smooth stemmed one, tall goldenrod, or Solidago gigantea, for those of you who prefer the Latin names. Annoyingly, as is often the case, some books use late goldenrod for this one and tall goldenrod for that one. Aren't common names wonderful, but I'll stick with this system. It's a model system for us. It's a really nice way to study this kind of thing because, first of all, the plants are abundant across North America, and if your first picture in your head when you say goldenrod is almost certainly in these species, so the abundant ditch, old field, disturbed place, and prairie things. They grow together in the same habitats, and Steve and I were this morning checking them out on Kaler and Kettlehole and what's the one I had? Morey that I hadn't been to before. Those two I've worked at before, but not Morey. And you know, they're there in the same places, in the same habitats, growing together all the time, so that's great. They have a ridiculous number of insect herbivores that attack them. on the order of 100 or more insects that eat one or the other or both of these goldenrods, not necessarily in any given field, but across their range. And each of those is attacked by its own parasitoids, which again I'll get to later if you don't know what that means. And so we can use the system to ask not just, oh, do we have an example of evolution through specialization, but does it happen frequently? We can actually try to assay whether it's a rare special thing or a common occurrence for an insect species to evolve a pair of specialists, one attacking each of these two available hosts, and to try to figure out what kind of ecology might control that process of diversification. I'm not going to successfully answer that question tonight, but I'll show you some sort of approaches to it. I'm kind of going to go back here and start with the simplest level question, and that is whether we can get the evolution of distinct specialists, one on late goldenrod, one on tall goldenrod, if we start with an insect that eats both of them. So can you go from a single generalist that takes either, and can you evolve a pair of specialists that eats, one of which eats each? That's a terrible sentence. A pair of specialists, one on each host. Okay, so that's sort of the question. How frequently do we go from generalist herbivores to pairs of new specialists? And I'm going to show you just one example of that. This is not that particular, so the swelling on the stem there is a gall, so it's an insect-induced plant tumor. And this is another one. There's a stick in here. This is either I should have asked you for that. I'll just jump. So, whatever. That is another gall species. It's actually a moth, and that's a little brown moth that makes it with the horrible name Oromachima gallisolovaginus. It is the goldenrod spindle gall moth. That gall is sort of spindle shaped, whereas those ones are a little rounder. So we asked, okay, we know that this gall occurs on both of our goldenrods. We asked, is it a single generalist moth eating both, or is it a pair of distinct host specialists? You can't tell them apart. To look at them, the moths that come out look exactly the same. The caterpillars inside the galls look exactly the same. So we have to do sort of some molecular work. And I'm only going to show you tiny amounts of this kind of data, but this is a tree for mitochondrial sequence, DNA sequence data for a whole bunch of collections of this moth from either tall goldenrod, which are coded in that pink and red are supposed to match, but they don't. Or from late goldenrod in the black here. The cryptic abbreviations are, well, there's Cone Marsh, which is near Iowa City, for example. 417 is near Ottawa, Ontario. They're from all over much of sort of central eastern North America. And the thing that leaps out at you, right, is that all the tall goldenrod moths group together inside the bigger tree. And what you infer from that is that these are genetically distinct from the others, first of all, or they wouldn't all group together. And second, that what's happened here is we've had the evolution of the tall goldenrod form from the late goldenrod form. So within the ancestral late goldenrod form, we've had colonization of tall goldenrod followed by genetic differentiation. And these things now are probably good biological species. We don't recover hybrids very often, if at all. They seem to be pretty well independent, even though they're morphologically cryptic, you can't tell them apart any other way. So, okay, yes, we can find that these things, that many insects that appear to be generalists attacking both of these things, this insect at least, is not actually a single generalist, but a pair of relatively recently derived specialists. More about the timing a little bit later. Now, it's not a weird thing, or lots of insects do that. So here are 4 different gall makers. And so this one here, the ball gall fly, that little fly makes the gall that I just passed around. This moth makes a different kind of fusiform gall. There's one I just talked about. This little fly makes that gall that sort of replaces the top of the plant, the inflorescence with a tight bunch of leaves. You may have noticed that as well if you've looked at goldenrod. And so these are 4 gall makers, and we say, okay, we'll assay each of those 4 and see, okay, how many of those are actually pairs of specialists as well. And the upshot is, I'm saying back to the camera a lot, aren't I? The upshot is that that spindle gall moth is not a weirdo, right? In fact, specialization is rampant in these insects. This is just 4. We've done other ones as well. But 3 of these 4, the ball gall fly is not actually our work, although we've had worked on it. These 2 are, and we've done other insects and found that it's actually relatively common to have the evolution of new specialists through changes in diet specialization through picky eating. So this is cool. So we know that this process is frequent, it's common, it happens a lot, and that's good. But it doesn't get us very far towards what actually controls it. And it's also worth pointing out that the evolution of new specialists is common, but it's not universal. So this moth here has not evolved a specialist pair. It's just one moth that eats whichever of the two goldenrods it comes across. So not everybody's evolved to be a specialist, and that's almost curious, because if everyone else could do it, why isn't this guy? And so we want to think about, okay, what kind of ecology controls when and how and whether a species diverges to form two specialists. So we can look for some clues in this system because we have multiple herbivores attacking the same pair of plants. So everything else is the same except for the species of herbivore. They all have the same opportunity to evolve pairs of specialists. Some have taken it, some have not taken it. And we can look at whether they've taken it in the same way, the same place, the same time, things like that. So just as a couple of examples, if you look at the direction in which they've expanded their diet, so you could have had a species that exploited late goldenrod move on to tall and then specialize, or it could have been a species that ancestrally used tall goldenrod, moved on to late and specialized. But in fact, 3 out of the 3 cases where we can actually determine the direction of the shift from molecular data, in all 3 cases the move is from late goldenrod, the hairy one, to tall goldenrod, the smooth one. So that suggests there's some predictability in the process. The things happen the same way every time. And that's true for the direction of the shift, but it's not true for the timing of the shift. This is the most complicated thing I'll show you tonight, so don't panic too much. This is an attempt to infer from that sequence, that DNA sequence data, the timing of the original divergence of the generalist into 2 specialist forms. So the way to read this is along the bottom, we plotted the percent divergence in DNA sequence, and most of you will know about sort of molecular plots. And we think mitochondrial DNA in insects evolves somewhere around 2% per million years, but there's a let's be cautious lower rate, and there's a let's be cautious higher rate. And so for each of the 3 insects that I showed you, that we know there are a pair of races, we can sort of ask, okay, this one, here's our spindle-gall moth, it's around sort of 2% or a little bit less, and so we can say, all right, that's about 1 million years, plus or minus some error, depending on whether our rate number is exactly right or not. So roughly speaking, on the y-axis here is your estimate of the time since the split. And the only important thing with the graph is that none of these intervals overlap, which means that we know that each of these has started this process of divergence at a different time. The bunch gall fly, I don't have a picture on here, these races are very old. Our spindle gall moths are quite old. The ball gall fly is very new. It's probably less than 100,000 years or so. So we have discordance in the timing. So it's not the case that everybody goes at once, because let's say the glaciers come south and the range of the planet is split up and you get an evolution like, well, the things are in different glacial refugia. This process can kick itself off at different times for different organisms. Why? That we don't know. But there's complexity here that's sort of interesting to get into. Let me leave that behind for a minute. Let me quickly ask something about parasitoids. Because you might think, well, okay, if herbivores can specialize on 2 different host plants, what about a parasite attacking the 2 different insects that are the 2 different host plants? So the kind of parasite we're talking about here are called parasitoids. Most people will know what that is. These are insects that lay their eggs in or on other living insects. The larvae develop inside the host and don't immediately kill them until normally when the adult parasitoid emerges and then the host is killed. So here is an ichneumon laying its eggs inside of the trunk of a tree on beetle grubs. These are eggs outside a caterpillar, and this is what happens when the parasitoid larvae are mature. They burst out of the caterpillar, and that is clearly fatal. If you've seen the movie Alien, that's what parasitoids are like. There aren't any that attack vertebrates, thank goodness. Most parasitoids are also moderate to extreme host specialists, and so you can say, all right, in our system, can we find specialist pairs of parasitoids exploiting the specialist pairs of herbivores? So this is one little wasp that attacks larvae of that spindle gall moth. There's an opened gall, And inside you can see what looks like it might be a caterpillar. And it sort of is. Here's a little bit of a blown up view. This is sort of the caterpillar's head capsule, but it's kind of all fat and stuff full of stuff. And with close up inside, each of these little grains of rice is a wasp larva. And so this entire caterpillar has been replaced by, these are actually all clonal offspring of a single egg, which is pretty cool. And this guy's toast. So it's a really neat system. Our interest was, okay, we know the spindle-gall moth, remember, has evolved these specialist pair. Is the wasp that attacks it also a specialist pair on the specialist pair of herbivores? And again, this calls for sort of molecular data because you can't tell them apart to look at. And here, sorry, the format's a little different than the other tree, but it's the same kind of idea where collections that group together are genetically similar and collections that are far apart are not. And here we've collected from 3 different sites in Fredericton, Minnesota, and Toronto. And in each place, you see that the collections from the same site group together, but the collections of 3 different hosts are genetically distinct. And this p-value, a low p-value, says these are genetically distinct. They're not, you're not sampling the same wasp off the 2 hosts. So the bottom line, if you don't like this kind of graphic, don't worry about it, is that Copodosoma, like its host, is in the early stages of evolving these specialist races. The distinction is very subtle in this wasp. So this is very, very recent development. And amazingly, this seems to be happening independently in several different places across the geographic range. And that to me is just mind-blowing. Not only is it easy enough that lots of species can evolve specialist pairs, but it's easy enough that the same species can initiate the process independently in different places across its range. That sort of blows my mind. And just like with the herbivores, we can ask across the whole system, okay, we have lots of different parasitoids. Do many of them evolve complexes of specialists? And the answer is, yeah, again, yes, they do. So I only have 4 on here, 2 of which have host forms, and 1 of which probably doesn't, 1 we're not sure. There are a couple others that aren't up here that also have host forms. So like their hosts, the herbivorous insects, the parasitoids also, by being picky eaters, are speciating and increasing biodiversity. So that's pretty cool. So so being picky on these 2 different species matters, and it helps. It's an engine of biodiversity. But if we go back to this field, sort of a halfway through sum up is, we've learned that herbivores evolve easily and frequently into complexes of specialists. Not all, but many do. Parasitoids do it too. So okay, specialization helps evolution of new biodiversity. But I lied to you about this field of goldenrod. It's not actually a field of 2 goldenrods, because it's more than, there are 2 species in there, but within each species there's variation too. And this is something I totally missed. For about the first 7 or 8 years of working in this system, I did what most people have done in the kind of field, and really focused on those 2, well, I'm going to call them totally distinct goldenrods I passed around. Some of you might be pardoned for thinking those distinctions were a bit subtle, but to me they're pretty marked and obvious. But in fact, there's more going on here. So here's another way that the goldenrods in that field aren't all the same. In fact, they differ in what we call ploidy. So I'm sorry, it's a mixed audience, so I know I'm insulting some of you and leaving some of you behind. I hope I'm sort of in the middle somewhere most of the time. All your cells package the DNA into chromosomes, and all of us in this room are diploids, 2 copies of each chromosome. So humans have 13 chromosomes, 2 pairs of each. 26 chromosomes, 2 of each. 26? 23? 26?
23.
Thought someone help me. Anyway, shouldn't have used that example, should I? But especially in plants, duplication of chromosome sets is really common. So lots of things have not 2 of each chromosome, but 3 or 4 or 5 or 6. Particularly even numbers are very common. And in the goldenrods, we find, in fact, that one species will have diploid forms, so 2 sets of chromosomes. Also tetraploids, which have 4, so tetra for 4. Hexaploids for 6. Goldenrods don't tend to go any higher, but many things do. Some of our ferns have 88 ploids and ridiculous things like that. Just unbelievable. And goldenrods, fortunately, 2, 4, and 6 are common. And we got interested in this, so we thought, well, that's interesting. I wonder if the insects care about these different ploidies. Because in fact, it turns out that that you can walk into a single field, at least around here, and the goldenrods will be a mixture of ploidy. Some will have 2 chromosome copies, some will have 4, some will have 6. The ploidy is heritable, so these, it's not just sort of accidental. The tetraploids will have tetraploid offspring and so on. So you've got sort of independently evolving lineages of plants within each of the 2 species in that field. So I'll talk to you about late goldenrod. That's the hairy one. Before flowering, it's really hard to distinguish the ploidies. The higher ploidies do have like larger flower heads and larger seeds, which is typical. A lot of our agricultural crops are high ploidy for that very reason, that they tend to have bigger fruits and bigger seeds and so on. And they co-occur locally. So in the east, we mostly have hexaploids. In the far west, mostly diploids. In this zone through the middle, we have both and also tetraploids. And Iowa is right in the middle of that zone, so we have very similar. Oh, this is great. We're still in Iowa at the time. This is exactly the right place to do this. So we'll see what's going on. So we actually asked how the insects responded to ploidy variation within that species. So here's a little transect across the state. The little pie chart showed the frequency of the different ploidies at each site, and all that matters here to you is that we had found many sites at which all 3 ploidies were present. So we can actually go out and say, okay, what fraction of the plants that are available to an insect are diploid, tetraploid, hexaploid? That's what the pie chart is. And we also go out and say, okay, what fraction of the plants that are being attacked are diploid, tetraploid, or hexaploid? Are they preferring or avoiding one of those ploidies? And we can do that for these. I'll show you data for, well, I won't. We did it for these 5 herbivores. I'll show you data for just a couple of them. And so there's sort of 2 cool things about science. And the first cool thing is that sometimes you can learn something no one has ever learned before. And so for a minute there, before you tell anyone, you know something that no one else has ever known before. And this is what happened here. So we go out there and we say, oh wow, the ball goes. The way to read this graph is that if a bar is above the axis, that's a preference for that ploidy. So if the insects use ploidies randomly, don't care about ploidy, then all the bars would be right along the line. A bar below is avoidance, a bar above is preference, and this ball gull, when you've got passing around, it loves tetraploids, really loves them. It hates diploids, hexaploids it's sort of iffy about. And no one had ever shown that before, and we thought, okay, that's really cool. So that's the first thing about science that's fun. But okay, we're learning something here. And the second thing that happens that's fun about science is that sometimes you learn something no one else has ever learned before, but you also don't understand it. And that's what happened next. Because these are data for the bunch gall fly, and it has really strong preferences about ploidy, but they're different every 50 kilometers down the road. So at, let's see, this is Johnson Sock Trail, I think, which is just over the border in Illinois. They love tetraploids. Here's Norton Nature Area, which is east of Iowa City? Northeast?
Northeast.
Northeast, anyway. Probably these might be 100 kilometers apart or so. And it absolutely hates tetraploids there. Over here it loves diploids. Here it hates them. This is Beaver Lake. This is, I don't remember actually. That's embarrassing. Okay. Western Iowa somewhere. So this thing can't figure out what it wants. It's a bit like me. I didn't know what the hell this meant, but we started thinking about it a little bit. I thought, well, how do we explain this? We know that herbivores might respond to ploidy in a couple of ways. They might be interested, or they might be responding to traits that are directly the result of polyploidy. So things like growth rate or cell size, where hexaploids, all hexaploids are different from all diploids because they have bigger cells, they grow faster, something like that. If that's the case, then all hexaploids are kind of the same, and the insects ought to prefer all hexaploids or not like hexaploids. We have to be consistent about it. But it's also possible that we could just be looking at preferences for plant lineages that are evolutionarily isolated by their different ploidy, because ploidies don't breed together. So the diploids and the hexaploids don't breed. And so since the origin of the higher ploidies, these have been genetically distinct and evolutionarily independent. And anything else that changes, anything else, any other kind of variation that arises between those lineages are things that the insects also might care about. But if that's the case, they're not really responding to ploidy per se at all. They're responding to some other trait that's evolved in that ploidy isolated lineage. And if that's the case, they might not be consistent about it. They might happen to like the hexaploid lineage here, but they might happen to really like the diploid lineage that evolved over there. Because these higher, I didn't mention the higher ploidies are also multiply derived, so you get high ploidies evolving from the low ploidies repeatedly in space and time. So what this suggests to us is this pattern, that the discordance is the common pattern. This suggested to us that in fact insects were probably responding not to ploidy per se, but instead we're just using ploidy as some kind of easily assayed marker for these lineages within species that the goldenrod that they're seeing as distinct and different targets. So that field of goldenrod wasn't just 2 species, and it wasn't just a bunch of ploidies. It was actually a whole bunch of genetically distinct lineages, a bunch of different genotypes, maybe hundreds, maybe thousands of them, all of which could be seen differently by the insects. Okay, so that just makes things more complicated, right? Why does that matter? Well, it's really interesting for 2 reasons, one of which I'll talk about, one of which I won't. I won't talk about this further today, but you know, the one thing you can sort of ask is, well, if we can get the evolution of specialists on Multissima and Gigantea, or sorry, tall and late goldenrod, the 2 I passed around, why couldn't we get specialization on finer genetic distinctions, on tetraploids or hexaploids, or on genetic lineage A or B? And we haven't followed that one up, at least not yet. But I don't see why it's not possible. We just haven't done that kind of work. The other thing, and the reason I sort of said this is a story of the sort of evolution of the way I looked at this field of goldenrod, is this realization about ploidy was what led me to think that I've been approaching this all wrong. And this was not just a field of 2 species. It wasn't just a field of, let's see, 2 times 3, 6 ploidies. That in fact, I should be caring about the thousands of different individual goldenrods in that field. Because the insects were noticing this stuff, even if I wasn't. And I started to think about maybe we didn't have to necessarily have host specialization on every single individual, but the way insects responded to variation among individual plants might be shaping the way they evolve or don't evolve specialists at the species level. And that sounded a little bit complicated, but I'm going to try to show you just a very quick sketch what I mean by that. I'm going to sort of, I think, skip ahead a couple of slides. Yes, here. Okay, and I'll go back later on. What I started thinking about was this sort of conceptual model where we think about variation out there in a field. And so imagine that you had a whole bunch of plants of one of those goldenrod species or 2 of them, and you measured a bunch of traits about them. I'm going to say we measured, so we have shorter and taller plants, and we have smooth and hairy plants. And so those 2 I passed around, one of them, I think the tall goldenrod, the smooth one was also rather large, so it would be both large and not hairy. It would be down here, right, this triangle. And the hairy one I passed around is a little bit shorter, so it might be sort of up here. So you have sort of this trait space that summarizes variation among all the available plants. And a fly might be flying around in the field thinking, oh, I like these ones, these big but not hairy ones, for example. And the triangles and the circles here are supposed to represent the 2 different species of goldenrod, which, as it happens, I pass them around, so there's the hairy one and the not hairy one. Okay? So what we're kind of imagining now is that if you look at how the insects respond to variation within a species of host, it might be something about how or whether they can shift to the other host and what happens after they do. So just as an example, I'll show you a couple of plots like this. This is a case of a hypothetical, as I often say, hypothetical insect, which is really a specialist on the little triangles, which in our case is, let's say, the not hairy goldenrod, the tall goldenrod. So you'll notice it's attacking a couple of the circles. This is making a couple of host choice errors. It's attacking the wrong species, but it's attacking individuals of that wrong species that are, well, they're almost right. They're very close to its real host. And this is potentially the way insects expand their diet in the first place, how they get to eat more than one thing in the first place. The specialist here can start eating the wrong thing by mistake. Once it starts accepting it by mistake, natural selection can act to make it better and better at finding and exploiting that resource. And so this potentially gives you a way to to take a specialist and actually make it into a generalist. Then we say, okay, well, what happens next? Well, can that generalist then become specialist, but a pair of specialists again? And here's sort of a potentially an intermediate stage. So you might get that shift, and so this insect might attack, the red ones again being attacked, might now, later in its evolution, have come to accept both species as perfectly good to eat. But it likes the most, in this case, tall golden white, it might like the most tall goldenrod like late goldenrods, and the most late goldenrod like tall goldenrods. You can see I had trouble saying that. And this makes sense, and so I've got descriptions of what that means, or they aren't terribly important. The notion here is that the individual level variation of the plants might matter, and here's a particularly interesting stage that we wonder if this is important. I say wonder because we don't know. But if you're going to take a generalist that eats both hosts, like this, and somehow have it evolve to become a pair of specialists that only accept each, somehow you have to stop these guys from mating with each other and from hopping back and forth from host plant to host plant. You have to set up some kind of evolutionary barriers between them. And how could that happen? One that could happen is if somehow their individual preferences got to look like this, where now they are attacking the least, here I'm going to go again, the least tall goldenrod like late goldenrod, and the least late goldenrod like tall goldenrod. They're taking dissimilar individuals. That can happen for a bunch of reasons, I won't go into in detail, but if it can happen, then these kinds of preferences should greatly reduce the number of host choice mistakes back and forth, and the number of mistaken matings between them. And that can happen in part because these things often mate right on their host plant. So once they've chosen the host plant to hang out on, that's where they find their mates. So this could reduce mating between them as well. So this kind of thing, if this pattern of individual preference could come up, then you have a path to an evolutionary barrier and therefore the evolution of two separate specialists from our generalists. So can this happen? Okay, that's her question. This is Chandra, and she's my PhD student. And I've done a crappy version of testing this hypothesis, and she's going to do a better one. So now that I've totally embarrassed Chandra. So I'll show you the crappy data. This is just one piece of data for one real, so the last triangles and circles were hypotheses. This is actual data. And you don't have to worry much about it. This is for the red goldenrod aphid. We don't know if it's a generalist or a specialist, or a pair of specialists. It's on both hosts. And here are a bunch of real data for real goldenrod plants. We've measured a bunch of traits and plotted them in some kind of fake trait space like that. Or I should say fake, highly simplified trait space. And you can see more or less this is actually mostly hairiness, so that's the late goldenrod, and this is size, which seems less important. So late goldenrod, tall goldenrod. And again, the filled red circles are the ones that are attacked, and the open ones are not attacked. And it's very difficult to see in this slide without squinting a lot, and the sample size isn't very good. There's a tendency here for the circles to be a little lower and the triangles especially a little higher up here than expected by chance. So there's a tendency, it's not significant with this terrible sample size, but there's a tendency towards that pattern I showed in the last slide. These things are actually preferring dissimilar individuals to the two hosts. So you gotta squint a lot to see how this one looks like that one. But if you squint just right, trust me, the sample size is poor. But this is pretty exciting because if, in fact, we can sort of demonstrate this pattern and then couple that with molecular data to show that insects having that pattern or achieving that pattern begin specialization, then we'll have our finger on a way that we can link that sort of very fine scale, sort of what seem like unimportant minor decisions that the insects are making about, well, I like that goldenrod better than that one right there. We can link that up to the evolutionary trajectory of entire species and the evolution of new biodiversity in the goldenrod insects. But by extension, we think this is likely important in lots of other things. That's okay. And so I wanted to sort of sum that up by saying that we have good evidence that specialization, I think, is a powerful force driving evolution of insect biodiversity. not an inexorable force. We do have some generalists left. And I think that's actually an interesting thing, that not everything is ultimately picky. And so we can think too about sort of evolutionary forces that might favor being a generalist and not evolving complex as a specialist. Just as one example, if you're a real specialist, you may often find your only acceptable food source going extinct. And if you're a generalist, you can always shift to something else. So there are certainly pressures in both directions. We think that part of the key to understanding this is thinking about these goldenrods, these fields of goldenrods, not as a pair of species, not as a bunch of ploidy levels, but as individuals. Lots of individual variation may matter to the trajectories of whole species, which means that field of goldenrods is a lot more complicated than it looks and a lot more interesting than it looks. This matters not just to insects and goldenrods. I study insects and goldenrods because they're a great study system. You would be forgiven for not thinking they're terribly important economically or anything like that. They actually are. I mean, in Europe, our goldenrods were introduced in our invasive weeds, and there's interest in using the insects to control them. But even here, not the goldenrod insects, but this kind of behavior where insects shift their hosts and then do or don't evolve specialization matters immensely to agriculture and forestry and things like that. For example, the shift of corn borer onto soybean is an example of exactly this kind of thing. For example, in forestry, we have a bunch of sawflies and moths that are major defoliators of our forest trees. Where I come from, softwood forestry is a huge industry. Spruce budworm comes along and devastates the forest. It's on black spruce, white spruce, and balsam fir. We want to know, first of all, is it actually the same insect in all three? Will the same control strategy work in all three? And secondly, do we expect it to evolve distinction? And is our management possible, or can our management strategy actually drive it to change its trajectory and evolve, either to evolve new specialist races or for existing ones to collapse? So there's lots of sort of applied angles on this in systems that are more difficult to study than our wonderful goldenrod system, where we can learn a lot that we can hopefully apply elsewhere. The last point I wanted to make about this stuff is that there's another angle that I think is really important about this system. It tells us, it teaches something interesting. That's not actually Steve, well, it might be. I guess there's a very small probability that the image I stole from the internet of a baby in the 50s actually is Steve Hendricks, but he does look pugnacious, doesn't he? Anyway, we're really used to thinking about evolution as a many millions of years thing. So I have his shirt too with the Tyrannosaurus on it, and there's the skull, and that's what, you know, if you say evolution to a kid, they say dinosaurs, right? Stuff that happened a long, long time ago. Darwin's book Origin of Species, published in 1859, and there's the other couple of timeline references, not to scale, right? Here's a few of these insects. This is the goldenrod ball fly, maybe 100,000 years ago. Here's the apple maggot fly. This thing began its process of evolving new specialists, speciating, new biodiversity, at the same time that Darwin was sitting in his cottage writing The Origin of Species. And this, I haven't talked about this bug. This is a silkberry bug that occurs on silkberry, a golden vine tree, golden rain tree. Darn it. Hugh Dingle's work. And this thing didn't start to diverge until the 1960s. And so this postdates Steve and is about coeval with me. And it has identifiable, genetically distinct, or actually morphologically distinct, I believe. I think there's some proboscis length variation here. So it has evolved a new pair of specialists in about 50 years. In other words, this stuff is happening around us all the time, like now. And new cases may have happened while we were talking here. And to me, that's really cool. So I like this stuff. And I believe, that's what all I wanted to say about the system. I packed a lot of different little studies in there, and I tried to give you sort of an overview of the thinking over the years and how it's evolved. So I may have left out a lot, I did do a lot of detail. I'd be more than happy to expand any bits that you find interesting or answer any questions you have. We've got lots of time if you want to take it. So thanks.
comment was that I was actually a much cuter baby, but we have to account for what happened since then. So, but sure, Steve will be glad to answer questions.
Sure.
So you said at the very beginning that all of the insect species you looked at had started on the late goldenrod and then eventually moved to the tall goldenrod. Do you know why they all started on the late goldenrod?
No, we don't. You might say, well, the tall goldenrod itself is just a recent origin. But if that was the case, you kind of expect that all the bugs would move sort of at once when that new species became available. That's not what happens. They show you the dates are different. I always have to admit the system isn't perfect. We have a rather poor understanding of the timing of the evolution of the different species of the host plant. In fact, even of the systematics of the genus. And so I don't know, for example, it may have been the case that late goldenrod was much more widely distributed. This is all rank speculation. And I'd love to figure out the easy way to get at that question, because it's really striking. I mean, it's only 3 out of 3, right? 4 out of 4, one I didn't show you, there's a flower beetle that lives inside those round galls, is also in that direction. So it's at least 4 out of 4. We'd love to know why. It's a very frustrating answer to your question.
I have a very simple question. Do you think the weather has anything to do with this? When I was thinking about your field of goldenrod,
That's a great question.
and a blast of wind going through, so does it make any difference to your results and things like that?
So at that level, whether I don't think so, there's all kinds of, that could absolutely set up variation in which individuals the bugs are on, but that variation then we wouldn't expect to be consistent from field to field or year to year. So we're looking for traits or characters that are associated with attack over and over again. But it's a superb question, because one of the traits that is important here is a plant phenology and insect phenology. So for example, in the spindle gall, the one I showed you the little tree for, we know the gigantea moth attacks earlier in the spring, just by a week or so, and emerges later in the fall. I've actually forgotten. Again, by a matter of a week or 2. And that's enough to help both target host attack and also keep the two, because the adults come out a couple weeks apart, that keeps them apart, that keeps them genetically isolated. What I think is important about that is that climate changes, and so if you sort of imagine this offset in phenology as kind of like a window that slides open and slides closed as climates change. And it may be that in order to start the process off, to start a new set of specialists, you need to have climates be such that for a long enough period of time, for, is it 200 years, is it 1,000 years, what is it, that the phenology is distinct enough that they can get started and specialize. And if the phenology window opens for a little while and comes closed again, and they haven't become distinct enough in other ways, then it just collapses back to a generalist. But if it stays open long enough, maybe it can evolve. And this could give you an explanation of why they start different times, because the phenology of each insect is different. The way it attacks the plant, the time of year it goes there. And so you could imagine the climate's changing in such a way that favored the evolution of new specialists in one species, but not in other ones. Right, so that's something we're really interested in, and we collect that kind of data on phenology and timing of attack. It's hard to collect data on thousands of years of the timing of attack, but it's certainly a really good suggestion. That was a good question.
Well, this divergence has been going on for 100,000 years approximately, or so.
In the ball golf flat, that looks like it.
Okay, now the valley that you were working in, is there different divergences going on in the next valley over, or the next?
For ball fly, it would appear that there is 1 or possibly 2 origins of the tall goldenrod form. Those data are Warren Abrahamson's data. He has published trees that look like the one I showed you, where all the tall goldenrod flies are in one group. There's this other funky little group over in the corner of late flies that don't seem related to the others. He's never really followed that up. So potentially a second origin. We don't have any evidence in our moth for multiple origins. You might expect coalescence. So it will long enough time, if you got multiple origins, you might expect them to hybridize and break down. That one parasitoid I showed you, we do have evidence from multiple origins. We've been frustrated trying to get better data on that. We thought some nuclear markers would work better, and it hasn't, but it looks like. I don't know in silkberry bug, for example, whether we've had that. Apple maggot's a little bit of a complicated story. There's some chromosomal inversion happened there in the south, I think, that fixed with the apple fly, and so that's probably a single origin, but it's not clear. So we don't know as a general issue. And to me, the idea that you can have these multiple parallel shifts across space is really exciting. The same thing happens in fish. So my postdoc supervisor, Dolph Schlitter, works on speciation in sticklebacks, and like many fish, stickleback tend to speciate, they invade lakes, and a single, in this case, is a marine ancestor, will invade a coastal lake, and then within a few thousand years, you'll have a lunetic or upper water column species and a benthic or bottom of the water species. This happens in parallel across lake after lake after lake after lake. So this process in fish is both frequent and predictable enough. You can almost say, okay, I'll put the stickleback in this lake, and you wait 5,000 years and you'll have 2 sticklebacks. And one of them will look like this, and one of them, the changes are predictable. The benthic is a bigger fish, it loses its armor, its mouth goes down, so the feet in the bottom, and the morphological changes are really parallel. We don't know as much about it in this case. I guess I rambled a bit there, sorry. This is so cool, I can ramble.
I have a question about the goldenrods are clonal. So when you're out sampling in the field, how do you deal with individuals that or do you collect the plant tissue so you know which individual stem it comes from?
We haven't actually done that, so you're absolutely right. So both species are clonal, and in a young old field, or a newly colonized, you can sort of see the clones, because you'll have seen this. You'll see a big circular patch. Oh, that's a clone. You can even see sometimes, you can see, even the prairies around here, you can often see that in one little area, the plants are just obviously taller, all the same color or something. That's a clone. But very often in an older field, you can't. They're all interdigitated. What we tend to do is kind of finesse it a little bit by, when we collect data, if we collect one here, we'll walk 2 or 4 or 5 meters before we consider collecting that one, so that we're trying to not bias our sampling by taking a whole bunch off the same clone. There's among clone variation, like that poly level stuff we're talking about, but there's also within clone variation. And one interesting angle on this is whether there's a difference in the importance of these traits, and this is some of Shanda's ideas, whether genetically controlled variation on individuals, the stuff that varies from clone to clone, might be more reliable and therefore evolutionarily more important in terms of preferences that lead to change compared to traits that are really plastic and vary within clones but don't have much information for the attacking insect. We don't know anything about that yet, but it's sort of down the road. The clonal thing is nice. It also lets you. It also means you can very easily pop them off the greenhouse and get many clones of a plant and do experiments. So it's a nice feature of the system as well. Anything else?
All right. Thank you very much, Steve.